Today we're talking about fetal development. So chapter 4, genes and chromosomes, right, in the DNA. So we have 46 chromosomes, and that's made up of 22 pairs of autosomes and one pair of sex chromosomes. Um, and so as the fetus is developing, that DNA, obviously, things that the woman, the female, um, will affect that development. So it's called teratogens, like the external environment, drug use, maternal nutrition issues, smoking, etc. That will affect that DNA development. So mitosis. So all of our cells, they reproduce through mitosis, like a replication, multiplication of those cells that have 46 chromosomes, and those are diploid cells. So mitosis, diploid cells, 46 chromosomes. The process of mitosis with sperm is spermatogenesis. The process of mitosis with ova is oogenesis. Now, once the sperm and the ova are um, replicated and they're ready, they go through a different division, which is called meiosis, where they become haploid cells. So they only have 23 chromosomes. Um, and so when it comes to the sex chromosome, the ova only have X chromosomes, sex chromosome, and the sperm have an X and some have a Y. So if you have a Y chromosome, you're male. If you do not, you are female. So the obviously, so then the sperm will determine the sex of the um, infant, because if you have one sperm that's Y and an ova that's X, that becomes male. If you have a sperm that's X and an ova that's X, then that's female. So once the fertilization takes place, it becomes a zygote. So the sperm enters the egg, that becomes a zygote. That process happens in the fallopian tube, and then the zygote travels through the fallopian tube into the uterus. And again, if you have one sperm and one egg, you get one fertilized zygote. If that splits in half, you have identical twins. If two sperm fertilize two egg ova, and now you have fraternal twins. Okay, so when the sperm fertilizes the ova, then a protective layer goes around it so that no other sperm can fertilize it. Okay, so and then also, in addition, there is some research coming out that the pH and the environment of the female reproductive tract, the vaginal canal, the pH can affect the survival rate of the X or Y sperm. So that's something that's kind of interesting. Okay, so the zygote was formed from the sperm and an egg, and then it's formed in the fallopian tube, travels down to the uterus. Um, oh, and then quickly, so inheritance. So I think one of the reasons we kind of talk about this a little bit is just so you get a little taste of the, dominant and recessive genes. And when we talk about different things in terms of various illnesses, for instance, things like, sickle cell. So sickle cell, in order to, like, someone can be a carrier but might not have it, right? Because it's one of those, there's a few of these types of conditions where you need a gene from each parent in order for you to express that condition. You can be a carrier but not get sick, but you can pass that on to a child. And if the child's other gene has it, then they can develop it. So there's sickle cell, there's cystic fibrosis is the other one. So when they do genetic testing with pregnant women, these are sort of the things they look for, that whether or not you have cystic fibrosis or sickle cell, those kinds of traits. And then I think it's interesting. I think genetics is very interesting if you want to look at some of that stuff. Okay, so the zygote travels down and plants into the endometrium. And that's where it then sort of implants, and you start to develop the chorion and the amnion. So the chorion, so that's developed from the trophoblast, which is the outer layer of the embryonic cells, um, and envelops the embryo and yolk sac. So this is sort of the outside layer. This is sort of the bubble that everything is encased in. And the zygotes and the fetuses, the embryo, they have yolk sacs. So the amnion is the second membrane, and it's a thin structure that envelops and protects the embryo. And so this is what will be filled with amniotic fluid, like a bag of water, the amniotic sac. And that's what the fetus then floats in. So the yolk sac forms, and that's sort of what nourishes all the, embryo and trophoblast. And then I think it dissipates after 6 weeks. One of its functions is the production of red blood cells. And then after 6 weeks, the embryonic liver takes over the production of the red blood cells. And then there are various germ layers, which then become various parts of the fetus. So the amniotic sac, the amniotic fluid, it maintains temperature, prevents the amniotic sac from adhering to the fetal skin, and allows symmetrical growth because the fetus just floats in there. So there's nothing inhibiting one side or the other from growth. They become buoyant and they can move, and then it's also a cushion. And the umbilical cord is also cushioned from injury. Okay, and so then again, the yolk sac lasts till 6 weeks, and it develops the red blood cells. And then these are the different germ layers, the ectoderm, which then becomes the skin and the hair and the nails, mucous membranes. And then you have the mesoderm, which becomes the skeleton, bone, cartilage, muscles, blood vessels, kidneys. And then you have the endoderm, which is the digestive tract, the respiratory tract, and bladder and urethra. So the 3 stages, so you have the zygote, that's the cell that's formed, the embryo, which is the 2nd to 8th week of development, and then so up to 2 months, and then the fetus, which is the 9 weeks till birth. And then of course, age of viability is 20 weeks now because of NICUs and all the technology that we have at our fingertips. Because I think it used to be 24 weeks. Okay, so accessory structures, obviously you have the placenta, right? So that is it's like a blood sac. It kind of looks like a tree, and that's that's on the wall of the uterine lining, and those blood vessels mesh with the mom's blood vessels, and that's how the fetus gains all of its nutrition, and that goes along the umbilical cord to the baby and the fetal circulation. So then the placenta, right? So you've got the fetal deoxygenated blood and waste products leaving the fetus through 2 umbilical arteries. And then fetal and maternal blood don't normally mix, right? Because you've got that barrier. But some things do cross the placental barrier. So, and things can, we'll talk about that. And then oxygenated rich blood and other, stuff is then carried through the artery, so through the vein back to the fetus. So there's 2 arteries and 1 vein. So again, the hormones, so you've got the progesterone, you've got the estrogen, and you've got the HCG. So that's that hormone that we test for in pregnancy, right? So when you think you're pregnant, you go pee on a stick, or they do, they dip your urine, or sometimes they draw blood samples and they're looking for that HCG level. Now, that doesn't mean anything except that so it's like a possible pregnancy test, right? Because we'll talk about that later, but yes, that's the pregnancy test hormone. And then the HPL, the human placental lactogen. Okay, so the umbilical cord. So you got the two arteries and the vein, and it's surrounded by Wharton's jelly, which, they're still kind of researching, but it cushions the vessels, you know, and probably nourishes them, probably has something in them that they need. Um, so the two arteries carry blood away from the fetus, and the veins return the blood to the fetus, right? Arteries go away from the heart, veins go to the heart. Same with. Um, okay, so then there's a couple of things with the fetal circulation, and I'll see if I can post like a video if you want to look at it, because it's quite interesting. Also, pages 68 and 69, you kind of get a breakdown of the fetal development. Um, okay, so the ductus venous diverts blood away from the liver as it returns from the placenta. Um, and then the foramen ovale diverts most blood from the right atrium directly to the left atrium. So if you remember the circulation in the heart, you go from the atrium to the ventricle, then back out, and then back to the atrium and the ventricle. Well, this is bypassing that, right? So there's a hole between the two atrium, and the blood is going in one and directly going to the other. So it's not going to the ventricle. Some does, but this hole allows some of it to be shunted. And then the ductus arteriosus diverts most blood from the pulmonary artery to the aorta. So the same thing, there's that little hole there. And so the blood doesn't do the full circulation. It kind of bypasses a lot of that. So the blood enters through the umbilical vein. About half goes to the liver. The remainder enters the inferior vena cava, which goes up. Through the ductus venosus, goes through the foramen ovale, and then the ductus arteriosus. So blood-containing waste product is returned through the arteries. Circulation after birth is like a quick adjustment, and this is where things can get tricky later on if these holes don't close up. But the foramen ovale closes within 2 hours after birth, and then permanently by 3 months. Ductus arteriosus closes within 15 hours, permanently in 3 weeks, and then the ductus venosus closes functionally when cord is cut, permanently about 1 week. After permanent closure, the ductus arteriosus and ductus become ligaments. So again, later on, if those don't close, that can be an issue. So fetal lungs, because they're swimming in the amniotic fluid, that's what they're breathing, and they're used to that liquid. So that's usually what maintains the lung expansion and formation. At birth, the reason vaginal canals are preferred than C-sections, other than other things, is because that squeezing through the vaginal canal helps expel some of that liquid. So that liquid should be gone when they cry. We kind of want them to cry sometimes to get that fluid out if there's any fluid remaining. So that's one of the things we assess when they're born is their lung sounds to make sure we're getting that fluid out. And then there are some hormones that play a part in that to help drain those lungs and prepare the baby to breathe air. So impaired prenatal development, of course, if the mom's not getting enough nutrition for the babies, it's called growth restriction. So there's uterine growth restriction, and there's other ones too. This is sort of a overview of some of the things. But so there's like small for gestational age and all these things that can be caused by smoking or drugs. But this is why we want them to do the prenatal vitamins and stuff to really get that nutrition. So the impaired prenatal development, so the undernutrition in the uterus can cause permanent damage, right? So if they're not getting that folic acid, they can have neurological damage that we'll talk about later. It's in the spine. They can have, they're just not developing. So, and then they can have other problems later on in life. So twins. So we talked about the identical versus fraternal, or monozygotic, dizygotic, right? Di two, mono one. So the monozygotic are the identical twins from a single ova, and then the dizygotic twins are the fraternal twins from two separate ova by two separate sperm. And to me, twins are fascinating. I have twins, and I just think it's really fascinating. If you really look at some of the research that's coming out, it's pretty amazing. So the twins, now usually if they're fraternal, they will have their own placentas, their own chorion, their own amnion. But when they're identical, you can have issues there because if they're sharing a placenta, if they're sharing amniotic fluid, if they're sharing all these things, one sometimes can dominate the other, and then one might not even survive or come out, frailer, weaker, smaller, malnourished, all these things. So identical twins, especially if they're sharing placentas and amniotic fluid, are watched closely, are monitored. So yes, so the two arteries and one vein, and the male sperm determines the sex. Okay.